So our last speaker before lunch is Dr. Jay Preston, who got his PhD from University of Illinois and has been spending quite a lot of years in the chromatography space. And so we'll be hearing about a new twin column technique. Thank you.
There it is. All right, well, thank you, everybody. Thank you for coming. I try not to keep between you and lunch for too long. This talk's a little bit different than the previous couple. Those were very modeling and theoretical and long-term. This is actually solving a problem that is in the development process. And it's a little more of a hands-on, like straightforward thing. Is that echo okay? Or should I be not closer to here? Okay, great. So we'll jump right in from here. So basically, this is the outline. I want to give you a little introduction to our Enriched technology. It is a twin column technology that's been developed in Switzerland and is now being implemented around the world. We'll talk about how the Enriched technology works, and I've got 3 case studies as a summary. For those of you who are less theoretical than the previous couple of talks, this is more hands-on. This is at the bench doing the studies themselves. So, trying to see which one's easier for me to read. Why would you even need to do impurity isolations? And that has to do with the drug development process. It works for biopharma, small molecules, peptides, oligos. Everybody needs to characterize the impurities that are left in their material at the end. So you need to identify those impurities, you need to have standards for your QC departments, you need to have sometimes activity testing. There's all kinds of good stories out there of some early development sample that was very effective, and then when they made or isolated or produced the material they thought was the active component in that sample, it was no longer active. It was actually some trace component that was in that initial sample that they couldn't find. This is a technique that can help you find that tiny little piece in the big pool of things. The other one is in the natural products world. They believe that some natural product, maybe it's cannabis, maybe it's something else, it's hard to tell, has components that are good for people. This will help you fish out those small impurities. So typically how that's done, and this isn't novel, this has been done forever with chromatography, is you start with a feed and then you run the chromatography on it, you collect a bunch of fractions, you do the analytics on those fractions, you see where your material is, then you pool all of those together and then you just repeat it over and over and over and over again. The concept of Enrich is really to get rid of the pooling, not to have to handle all those volumes and all that material as you're pooling things as you're going along, and to minimize the fraction collection and the analytics. So we want to optimize that vicious cycle of doing something a zillion times. And this is the traditional way of looking at it. So basically, it's a whole series of chromatographic runs that they're collecting a bunch of fractions, and then they do the analytical. So if you run 10 runs and collect 10 fractions apiece, you now have 100 samples to look at. Somebody in QC's got to run and look at. The automation's getting better, but it's still quite a bit of work in order to do this. So in the Enrich, and we're going to talk a lot about how the Enrich works. The lower left corner, it's a 2-column system, has column 1 and column 2, or the left and right, depending on how you're looking at the instrument. And basically what it's doing is you run a normal chromatography run on the first column. Everything that you want to collect, you send to the second column with a little more material. And then you run a chromatography run on that. Everything you want to collect, you send back to the first column, add a little more material, and you keep going back and forth as you're building on the column the components that you're trying to retain. And you run that for as many of these cycles as you want until you acquire as much material built up on the column as you need. And then you can basically change the elution profile and run a better separation on the material that's been retained on the columns themselves. So you're only fractionating once. So instead of having 100 fractions to look at, you may only have 10, is the basic principle behind it. And part of what it really does is it minimizes this middle step, that concentration step. You don't have all these fractions that are super dilute. You have much lower numbers of fractions that are far more concentrated, and it's a little easier to deal with, and you don't have to have that time-consuming, labor-intensive step of the dry down and potential loss and degradation and all the things that go with it. So it's going to save time for the whole process. Sometimes it'll even eliminate that entire step. So the Enriched technology delivers higher purity in less time. This is a publication that was put out, I believe it was last year, that compares a process for a MAB isoforms. They used some HPLC to isolate these different isoforms, and here they compare how much time and effort it took with the traditional HPLC, with the FPLC model, and then the Enriched model in order to obtain their materials. And then I'm gonna go a little more detail on how the actual Enriched process works. So basically it starts, this is the overview of the three-step process to it. Yeah, you start with chromatography, and it doesn't work with bad chromatography, so you need some separation. If your material's not separated, then really no chromatography is gonna help you get it pure. But once you get your chromatography developed, then you select the region within that chromatogram that has the material that you're looking to isolate. You then run the Enrich process itself, where you're concentrating the material that you're looking at on the column until it's at a suitable level. Then you take that material and then you elute it and fractionate it as it's coming off, and then that's the material that you can go and look, and you can pool out of those, or sometimes you'll get in a single pool what you're looking for, if your separations are good. So this is a slide that kind of goes over some of the case studies that we're going to look at. Mostly they're ion exchange case studies that we're going to be on. The equipment that does this technology, we call it the Cube. That's the benchtop version. There's a Cube 30, which is 37 mils a minute. There is a Cube 100, which will do 100 mils a minute. They'll run at 100 bar, so you can use steel pre-packed columns at reasonable flows, or you could use glass hand-packed columns also. It works in almost any mode that you really want to run. If you want to run ion exchange or affinity or reverse phase or any of those. You can run normal phase, but you got to be a little careful with the solvent choices for the compatibility. Some of the initial seals are not compatible with like straight normal phase materials, but you can get compatible materials to put into it. The detector, or the Cube itself, comes with two in-bed UV detectors, conductivity detectors, a pH sensor, and then there's an optional external variable wavelength detector if you want to do something that doesn't light up at 280 or 300. And like I mentioned, the 100 bar pressure. So this is kind of not quite a cartoon, but it's sort of like a step-by-step version that hopefully by the end of today you'll all be experts and you can explain this to your friends at home or your family if that's easy to do. Oop, wrong button. Okay, so there's a pictorial graph, actually two of them on the right-hand side there. So what we're really interested in this example is that kind of pink, light red, pink material in the middle. The front material are the early eluting impurities. The highlighted section there, that's the material we want plus some of the impurities. This is the only section that our target impurity is clean, in the center, and then we have some overlapping late impurities, and then we have late impurities without any of the material that we're interested in. So this process would be for if you're looking for one thing. And I'll show you some other ways to think about it if you're looking for a wide range of things in an area or a little more targeted. So we start with the feedstock in the first column. The feedstock goes through, and then we get rid of the waste, the early eluting waste material, plus maybe the edge of the part that we want. Then we put the 2 columns in series, we send the material that we want over to the second column, and it's okay if it's got a little bit of junk in it from the front too. It depends on how clean you want it right off the bat, or whether you want to do a little extra chromatography as you're doing the elution. So then we recycle the product, we get rid of the late eluting impurities to waste, we add more material to the second column so that we have fresh of the material we want and the things that we don't want. Is here, that's what this part is the feed. That part goes through, we get rid of the early impurities from the second feed. We reconnect the 2 columns, and then the more concentrated version of what we want goes into the first column. We add more material to it. We get rid of the late eluters in the second column. We add more material to the first column, and it does basically the same thing again. And then it just goes back and forth between the two, of isolating the region of the chromatography that you want and getting rid of the pieces that you don't want, and the entire cycle completes. Then you elute the enriched material that you want. You could have sent that material to the other column and then not added any more feed and run a different set of gradient or a different isocratic conditions as more of a polishing step. If you wanted a really long, slow, flat gradient to take care of that thing, that's another option that you can do. It really allows you to do a wide variety of different tasks. All right, so now that was if you were looking for just one target. We were really just looking for that middle peak. Let's say you wanted to know a whole bunch of things. You wanted to know all the impurities in the early and all the impurities in the late. So what you're really trying to do is get rid of the big peak, the big one in the middle that's overlapping everything else. So this little slide is a much more condensed version of that entire sequence that we just looked at, where the right-hand little blue purplish triangle, that's the weak retained or early eluting peaks. The big blue one is the product or the main peak, and the S for the yellow triangle is the strong retained or late eluting peaks. The middle upright bars, and this would be easier with a laser pointer, but I'm not sure the people on the video would be able to see it, but that's the section we're actually going to get rid of. So in the accumulation step, we're adding material, we're getting rid of the center cut, the peak itself. That's why in the next sort of set over, the W triangle and the S triangle are getting bigger as those are accumulating, because we keep getting rid of the, I guess we have a laser pointer right here. So we're getting rid of this section right here. These are growing because this one is going down as the ones that we cycle back and forth accumulate until we get to a point where we think there's enough of those materials on the column, and then we run our separation. And then as we run the separation, as the weak retaining material and the strong retaining material elute, we do a fractionation. So we've basically taken a large amount of material, crammed it onto the column, and then eluted it off as a single plug. This is an actual data example of that same principle. You can see that at every injection, the material that we're trying to obtain grows. The reason that the big peak doesn't grow is because we keep adding more to it. So it stays at pretty much a constant level, so the chromatography remains effectively the same. But what is growing are these impurities at the front that we are trying to collect. So as you run each different injection, the minor impurities get larger and larger and larger until it gets to a point where we think we have enough, and then we'll do the elution and the fractionation. All right, which leads us into our first case study, and this is obtaining a single component out of a dilute, or a dilute single component out of a mixture. The peak that we want is this peak. Now, why in particular they wanted that peak, I don't recall because I wasn't part of that project. But it would have been nice if they had picked one of these bigger peaks at the end. It would have been a whole lot easier to do. But of course, they wanted the tiny little peak right in front of the big peak. And what happens when you start loading more and more material is the big peak gets super wide and it encompasses the little peak. So you can't do a high load separation because your chromatography starts to degrade. So this was the results of the batch chromatography. This was kind of about as good as they could get. Honestly, I think they could have loaded a little bit more, but if they loaded more, it would have been overlapping with that peak right there as they were going through. But when they did the Enrich, and they ran one run at the end without adding more material so that they only caught the early peaks, now they have like this giant peak relative to other things. And the results is that for the Enriched, they were able to obtain material at 80%, and the concentration of that final product they got was 10x over any of the batch fractions. Now, they could have collected 10 batch fractions to get a similar amount of material, but the purity would have been low, and then they would have had to concentrate that down. So if you're looking to isolate, a single peak out of like a big forest of things, I mean, it's really pretty interesting technology. Which leads us to our second case study. This is looking at multiple impurities out of the mess. This is the chromatogram. This work was published recently, February of this year. It's an angiotensin II. This is the component that they want, and quite honestly, it doesn't look that junky. 92% pure, so that's pretty good by most people's standards. Not everybody, but most people. But they needed to characterize the little peaks because they were going to take it to the government to be able to sell it. This is a blown up version of that same chromatogram, and what they're interested in are all these little peaks. Actually, everything above 0.1% is what the government tends to say. This is the results from the Enriched loading, and you can see how all the peaks are growing as they keep running cycle after cycle after cycle, pulling out the main peak before they add more material into it, so it's only accumulating the little peaks as they're going along. And this is the chromatogram of the final one of those runs. Now all of a sudden it looks like we have giant peaks, and that looks a little more approachable from an isolation standpoint. And this is a comparison of the initial feedstock. These are actually the chromatograms of the individual fractions they collected. These are like where the fractions were. Some of them overlapped a little bit because the fraction collector couldn't always precisely do it, but that if we wanted to rerun those on a slightly different method to pull them apart, we could take this material and isolate a little further. But for the most part, this is good enough for what they were looking at. And the results, they did 18 cycles. They processed 38.2 milligrams of the initial material. It took them about 27 hours to do the entire loading of that material. It probably could have been faster on a bigger diameter column, maybe even a little longer to get a little better separation, but this was just the project that they chose to do. And they were getting, pretty decent main peak purity. Main peak means the target one. So like this one has a couple of them in it. This one out here has, like these 3 things in it. This one has a number of them right there. So that's like the main peak in each of the fractions. And the amount of micrograms of material they were looking for. So this was purely an identification project. They just needed enough material to do some identification work. Which leads me over to the case 3, which is the purification of some isoforms of a mAb, another thing that tends to be a little challenging. So this was a different variants. They have the acidic isoforms and the lysine variants. All the acidic ones elute early, all the lysine ones elute late. And these were in the feedstock, the percentages. It was actually pretty junky, in my opinion, if that's what they were looking for. Maybe that was by choice, sample in order to have enough material to isolate these, because now this isn't just for identification. They actually want isolated material that they could use as standards for the future. So they wanted to obtain enough material that they could retain it for their QC labs to have as marker mixes as they proceeded down their development road. So the first step is to find where they're going. They did this one in steps, and part of it is because some of these big ones are pretty big. They may not have had to do Enrich to really get a lot of material. They probably could have fractionated those as they were going along, as they were enriching this front part, sending that piece from column to column to column. They could have isolated these with a fraction collector on the fly. I mean, that's all within the capabilities of the instrument itself. And this shows this is actually the same data that I showed earlier that has the two different columns, and it's monitoring the progression. Each column, that's why there's two detectors in the system, is each column has a detector at the outlet, so you always know what's going on from as you go from one column to the other. They ran 5 cycles of the accumulation for this particular project. And then once it was accumulated on one of the columns, then they ran a flatter gradient version to pull those apart a little bit better so that they could cut a bunch of fractions across. These are some of the analytical QC that they did, and notice some of these eluted after the main peak. That's why they were collecting it as fractions, not part of the accumulation enrich. The ones in the front, these were the ones that were collected on the enrich process itself. And the results. So they were able to get milligram quantities of the different isoforms as they were running those 5 cycles, the amount of enrichment from the initial stock solution, may have worn out it's okay. We're just about done. With that, we may have, they have pretty good purity from the different fractions and the percent of the material in the initial load. And when you're running these kinds of things, you can actually do all kinds of statistics and different ways of evaluating and comparing the batch with the enrich. And in almost every case and everything, the enrich does so many different things. So to summarize where we're at for here, it's really a technology we like to say that it's fully automated, but there's a little bit of, you have some development and you have to be there for it to start, but it does run overnight. You can, it's got loading pumps and fraction collectors, and if you're comfortable leaving liquid flowing in your lab overnight without somebody there, then as most of us do, then you can let it go. It reduces the processing time. It definitely reduces the amount of fractions that you're looking at and holding on to and trying to boil down or dry down to get it operational. It's gonna reduce your QC load. You don't have to let your analytical run overnight or over the weekend to run these samples. And it's gonna save that concentration step. One thing we do offer, because not everybody has a need for a full-time system or things for this, they just have a once in a while, some sample they do. We do offer a contract services for doing this kind of work. We have a lab just outside of Boston and one just outside of Los Angeles that are equipped with these things that we can work with people to do impurity isolations and other things too, but that's what we're here to talk about today. So basically we use the technology, we do the isolation, the characterization. We send things back to people. We lyophilize it down usually. Most of the samples are water-based. We could rotovap if they were normal phase, but it just simplifies the tasks. It avoids the risk of degradation. You don't have fractions sitting on the bench for days or hours and hours and hours as they're waiting for the other fractions to collect. Pretty fast turnaround times. The overall workflow, is to establish the analytics, and that's actually one of the key parts. If you have good analytical work, that makes the enrich isolation much easier. If it's marginally worked out, it makes it a little more difficult. It takes a little more time and effort. And then we'll purify the standards with the Enriched. We'll verify it by an analytical system and then return the samples to people with a report. It does a lot of good things. I want to leave some time for questions and get you guys off to lunch at a reasonable time. Contacts in the US, you can contact me. In the Europe, Thomas, who's giving a talk today, I believe at 2:30 or 2 today. And the prerequisites are, we do a lot of confidentiality agreements and material transfer agreements. We do these like weekly with people. So, very familiar with how pharma works in that standpoint. And we'll use some self-developed protocols if your protocol needs some help, or we'll work with you to develop things. These are the sites that we have, and different addresses and things. We have a website, and I'm sure all of you can find like that. And with that, I'll save 5 minutes or get you guys off to lunch in a hurry. And thank you.
Thank you, and very nice talk. 2 quick questions. How do you deal with the binding capacity of the product on the chromatography media? When you're dealing with trace impurities, that's not an issue, but if you're looking to enhance something that's major in the feedstock, how do you deal with that? And the second question is, how much tolerance is there between the 2 columns that you need for, say, asymmetry, plate counts, and those types of things?
Sure. I'll start with the second question. So the variability between the 2 columns can be a reasonable amount. There's a tracking mechanism that the trigger is based off the detector. It's not purely a time. As long as the peaks shift more than like really change resolution, as long as the resolution is quite comparable, then the UV detection will track where you want it to go. The other one is the binding capacity and how that works when it's the major. So this Enriched is really a subset of the contachrome twin production material for isolation of oligos and peptides and proteins and things like that, where they do overload everything. And it works spectacular with affinity chromatography. But if you get into more of a partitioning mode, then you have to not run so overloaded for that binding capacity to be, way out there. It has to be a little more predictable and consistent. As long as it's consistent and the chromatography is worked out, then the software and the hardware will keep up with it. Okay. Yeah.
I was just wondering.
He's got the mic. You can go next.
It strikes me that you could also operate this in a different way where you use displacement chromatography in a second mode. And if you use displacement, you could get 100 to 1,000-fold enrichment. Massive, massive.
Absolutely. If you're using the front one as almost a screen and the second one to trap, and then you displace it, we call that a different technique, and I think that's like an integrated batch technique, where the 2 different columns and the modes are separate, and the second one's basically a trap. So you're doing a separation and then a trap and a lute. I guess you could. If it could be the same, as long as you don't run the wash aspect on the second column and let it accumulate. I couldn't hear that one. Yeah, exactly. They've done this. Like I said, this isn't brand new things. Putting it all into one box, having the software to do it, and having the predictive wizards to take your initial batch run and to develop the process, so you only have to do minor adjustments. That's where the new technique has really come in to give it the power. Yeah, go ahead.
Actually, I'm just wondering about the scalability of the technology. Up to which scale you have demonstrated that it works?
I think they have run it. on a 50 millimeter column at 100 mils a minute. So they're doing, what would that be, like a gram per run? I mean, somewhere in there.
Okay.
But
then potential do you see possibility of scaling up?
Absolutely. So when they do it for the main one, for the production, they scale that to the 20 to 40 liter a minute system, the 2-column system where they're isolating like the main peak out of things. And you tend to not overload like that for the Enriched because you're trying to keep the separation to get the little peaks. But you should be able to scale it as well as your chromatography scales from the cross-sectional area of the column.
Okay, thanks.
I just had a question on the fraction collection. Do you have the ability to collect on the basis of, let's say, when it hits a certain UV or pH or something, or is it just a dumb one where it just collects on volume?
It can collect on time or volume, but it can also be run off the detector. So it's like the slope of the detector above a certain threshold. It can collect on the upslope or the downslope of where it starts or stops.
Okay.
One more question.
One more, 15 seconds. No, just kidding.
I'll be quick then. Do you ever see issues of degradation or say shear degradants, aggregation from running the multiple cycles?
Not that I've seen or heard of. With some of the proteins, in the pH, depends on what pH range you are on for your proteins, because or peptides also. If it's in an area where the peptide or protein is known to aggregate, the more you manipulate it, the bigger chance there are for those. But as long as you're in an area it's freely soluble, it should not be a problem. That's a little more of a chemistry issue than a chromatography piece. All right, anything else? All right, well, I'll let you guys get to lunch.